Chromosomes are made of DNA. Each chromosome contains thousands of genes. The bases of sequence in a gene form a code that tells the cell what protein to produce. Hi, and welcome back. In today's science episode, I will explain the relationship between genes, DNA, and proteins.
Genes, DNA, and proteins. The genetic code. The main function of genes is to control the production of proteins in an organism's cells. Many proteins serve as enzymes that control chemical reactions in the cell. Proteins also help to determine the size, shape, color, and many other traits of an organism. Genes and DNA. A DNA molecule is made up of 4 different nitrogen bases. Adenine, that is represented by the capital A, thymine, represented by T, guanine, represented by G, and cytosine, represented with the letter C. These bases form the rungs of the DNA ladder. As you can see in the illustration, this is an DNA molecule, and these are the nitrogen bases. A gene is a section of a DNA molecule that contains the information to code for one specific protein. A gene is made up of a series of bases in a row. The bases in a gene are arranged in a specific order. For example, A, T, G, A, C, G, T, A, C. A single gene on a chromosome may contain anywhere from several hundred to 1,000,000 or more of these bases. Each gene is located at a specific place on a chromosome. Order of the bases. A gene contains the code that determines the structure of a protein. The order of the nitrogen bases along a gene forms a genetic code that specifies what type of protein will be produced. So this combination of the nitrogen bases forms a code, and they will know exactly what type of protein they should produce. In the genetic code, a group of 3 DNA bases codes for one specific amino acid. For example, the base sequence CGT always codes for the amino acid alanine. The order of the 3 base code units determines the order in which amino acids are put together to form a protein. How cells make proteins. The production of proteins in cells is called protein synthesis. During protein synthesis, the cell uses information from a gene on a chromosome to produce a specific protein. Protein synthesis takes place on the ribosomes in the cytoplasm of a cell. And here you have a picture just to remind you where are the ribosomes located. The role of RNA. Before protein synthesis can take place, a messenger must first carry the genetic code from the DNA inside the nucleus into the cytoplasm. This genetic messenger is called ribonucleic acid, or RNA. Although RNA is similar to DNA, the two molecules differ in some important ways. So we're going to study how they are different. Unlike DNA, which has 2 strands, RNA has only 1 strand. That's the first difference. So here you have DNA, it has 1 and 2 strands. And RNA has only 1 strand. Next difference. RNA also contains a different sugar molecule from the sugar found in DNA. And another difference between DNA and RNA is in their nitrogen bases. Like DNA, RNA contains adenine, guanine, and cytosine. However, instead of thymine, RNAs contain uracil, like you can see here. That's another difference between them. Types of RNA. There are several types of RNA involved in protein synthesis. Messenger RNA copies the coded message from the DNA in the nucleus and carries the message to the ribosome in the cytoplasm. Another type of RNA, called transfer RNA, carries amino acids to the ribosomes and adds them to the growing protein. Now, let's understand the protein synthesis. Basically, I wrote it down in 4 steps for this process. So, to make proteins, messenger RNA copies information from DNA in the molecules. Messenger RNA production. In the nucleus, a DNA molecule serves as a pattern for making messenger RNA. The DNA molecule unzips between the base pairs, and you can see this in the illustration. When I say unzips, it's like if it opens up, like you can see here. And what happens next is that the RNA bases match up along one of the DNA strands. They get together. This is the RNA, and this is the DNA. They get together. And the genetic information in the DNA is transferred to the messenger RNA strand. The second step. Messenger RNA attached to a ribosome. When the messenger RNA enters the cytoplasm, it attaches to a ribosome, where production of a protein change begins. The ribosome moves along the messenger RNA strand. The third step. Molecules of transfer RNA attach to the messenger RNA. The bases on the transfer RNA reads the message by pairing up 3-letter codes to bases on the messenger RNA. For example, you can see that a molecule of transfer RNA with the bases AAG pairs with the bases UUC on the messenger RNA. This was the illustration that I had before. Here, they pair up like this. The molecules of transfer RNA carry specific amino acids. The amino acids link in a chain. The order of the amino acids in the chain is determined by the order of the 3-letter codes on the messenger RNA. The last step. The protein molecule grows longer as each transferred RNA molecule puts the amino acid it is carrying along the growing protein chain. Once an amino acid is added to the protein chain, the transfer RNA is released into the cytoplasm and can pick up another amino acid. Each transfer RNA molecule always picks up the same kind of amino acid. Now, what happens if during all this process there is a mistake or something is missing? Well, in that case, mutations may occur. Mutations can cause the cell to produce an incorrect protein during protein synthesis. As a result, the organism's trait or phenotype may be different from what it normally would have been. In fact, the term mutation comes from a Latin word that means change. Effects of mutations. Whether a mutation is harmful or not depends partly on the organism's environment. The mutation that led to the production of a white kangaroo would probably be harmful to an organism in the wild. The kangaroo's white color would make it more visible, and thus easier for predators to find. Helpful mutations, on the other hand, improve an organism's chances for survival and reproduction. Antibiotic resistance in bacteria is an example.
I hope you found this video useful. Thanks for watching and until the next one. Bye!